Morning, Erez Lieberman Aiden, who has come here direct from Heathrow to talk about how the genome folds. Thanks very much, Erez.
Cool. It's a tremendous pleasure to be here. I'm going to be talking about some work. Unfortunately, I think some of you have already heard this talk, which I apologize about. There's a little bit of new material, but just kind of wanted to give people a big picture of some of the things that my group is thinking about. And then a pitch to try to get some postdocs. So anyway, I mean, this is just kind of very generic problem, right, that we're all interested in here. Human genome is stretched out. It's about 2 meters long. It folds up to fit inside a nucleus, about 5 microns wide. Obviously, it's folding up very, very extensively. In order to do that, there's very detailed pictures in the various textbooks of how this takes place. But the truth of the matter is that a lot of these intermediate scales between, this notion of DNA winding around histone proteins and sort of the notion of like a metaphase chromosome, there's a great deal of uncertainty. And so what we do is sort of explore this nether region. Now, one way that you could try to figure out how the genome is folded is to just look at things under a microscope. You can make them light up in different colors, green and red. And so if you light up some part of the genome in green and then some other part of the genome in red, you can get some general sense of how far apart they are. And this picture gives you roughly a sense of the state of the art. You can only look at a handful of sites at once. It's a little bit blurry. If you're very good, maybe you can do 5 different colors, right? This is roughly the state of the art in this space. Now there's this alternative sort of technique, and it really emerges from a very, very simple molecular biology piece of molecular biology called ligation. So there's this enzyme called DNA ligase, and what it likes to do is when it sees 2 pieces of DNA that are complementary to one another, say you've got some complementary overhangs, right? It sees this break in the backbone, these 2 missing pieces of the backbone, and it will seal them. It will ligate them. And this trick was invented, was initially discovered by the Hershey group back in 1966, and within a year, people were using this to do physics. And so there's been all kinds of efforts, towards using cyclization, as it was called early on, to try to understand the physics of DNA looping, the bendability of DNA. And there have also been efforts to apply this in vivo. Now, the first such effort was developed by Catherine Cullen and others in 1993. The basic idea is that you use ligase to join spatially adjacent DNA sequences to create chimeric sequences, and you could count the chimeras associated with each sequence pair. So if 2 sequences are nearby, you ligate them, and then you see how often this happens, and that tells you roughly how close they are. So you're using contact probability as a proxy for 3D distance. So the initial technique was actually very limited. What you do is look at one site in the genome and other nearby sites and ligate them to one another and see, get some sense of their spatial proximity. This was very substantially improved by Job Decker and others in 2002, essentially modifying the PCR strategy, introducing formaldehyde cross-linking. All of a sudden you could do better with this. In particular, you could start to sample things that were very, very far apart. So any 2 pieces of the genome that you were interested in. So if you think about the genome, this genome by genome matrix of possible things that could be bumping into one another, all of a sudden we can sample any entry of the matrix. So folks improved this further. Circular chromosome conformation capture allows you to sample a row or column. Chromosome conformation capture carbon copy allowed you to sample little submatrices. So this was good, and it was kind of very clear, when I was in grad school, if you just looked at this literature, where all this was going to end up, right, is eventually someone's going to just figure out how to develop an all-by-all technique. So I was like, well, how are we going to do that? That seemed hard. So I thought to myself, well, if we could do that, what would we call it? It was kind of very clear what the naming trend was, 3C, 4C, 5C, so maybe we'd call it 6C, or maybe if we felt really good, we'd call it 7C. But I thought, the critical insight was, look, if you had such a thing, you'd never need a higher number again, so I decided we'd call it high C. Anyway, so it had a zippy name, so I thought, well, this is clearly worth working on further. So the basic idea is, right, if two pieces of the genome are physically in contact, we want to figure out that they're physically in contact, and we want to be able to do this everywhere in the genome all at once. So the way that you do this is you start by cross-linking DNA using formaldehyde. This is essentially kind of like, shooting little, bits of hot glue through the nucleus. Things that happen to be in close physical proximity will get stuck together. You can now cut them with a restriction enzyme. So you cut the genome into literally a million little pieces. Some of the pieces are now stuck together. Here's where this is really the only step that deviates from previous techniques, and kind of actually this is the step that really gives this technique some power, is that you take these overhangs. So restriction enzymes will cut at particular defined sites in the genome. What you do is you take these overhangs and you fill them in. You add a couple of bases, and one of the bases is biotinylated. Right? Biotin is this chemical moiety that has this interesting property that it binds very tightly to another chemical moiety called streptavidin. And so what that means is that if I ever want to fish out things that have biotin attached to them, I can do this very, very reliably. And this is something that we're going to take advantage of later. So let's just take stock of where we're at. We started with two pieces of DNA that happen to be in physical proximity in the context of a much larger genome. And we've now locked in the two kind of local fragments, two local little snippets of DNA that encode that information. So now we introduce the DNA ligase, and what we've done, right, is we've gone from two things that happen to be in physical proximity to a circular piece of DNA, which is, in some sense, the receipt, right, the smoking gun, the proof that these two things were in spatial proximity. Because now the two sequences have come together, have ligated, and have formed one circle. And the only reasonable way that this circle could have formed is this sort of process, right, which is that these two sequences were in close physical proximity. So now it's just a sequencing problem, right? And we're very, very, very good at sequencing. So one thing that you want to do is, there's lots of boring pieces of this, and it's very advantageous to get rid of them, right? If you just read pure blue sequence, that's uninteresting. Right? It doesn't tell you anything about physical proximity. You just read pure red sequence, that's also uninteresting. It tells you nothing about physical proximity. What you want is these places where the red meets the blue. That tells you what two sites were in contact. So you can shear this, chop it into little bits, and pull down with biotin. And so you'll get some of these biotinylated bits, which represent the contacts, and then the rest of them you lose. Now, in principle, you can actually do this without that step by doing size selection. In fact, some folks are now doing this protocol in that way. And that's totally reasonable. I think it ultimately comes down to a financial calculation, basically. If you include this kind of step, then you can get, say, 80% of what you're reading is contacts. If not, then about 20%, say, of what you're reading is contacts. And this actually makes a big difference when you can spend $50,000 on such an experiment. So you'd rather have 4 times as much data. But anyway, you put this on a paradigm sequencer, and you're just reading off lots and lots and lots of these contacts. So anyways, it's kind of clear how this works. Are people on the same page? Great. So just a couple of disclaimers about how this works. This is an ensemble average. So we're looking at 20 million cells and all the contacts kind of aggregated from them. So that's something one has to be really, really careful about. Obviously, ensemble averages can really misrepresent things. If I were to, for instance, take the ensemble average of the people in this room, I would conclude that the average human has 1 ovarian, 1 testicle. That would obviously be a misrepresentation of reality. So we have to be very, very careful and cagey about how we treat ensemble averages. The second is that this isn't measuring distance. There's a humongous temptation to just say, well, contact probability is pretty much equivalent to distance, and therefore we have these pairwise distance matrices, and therefore we can essentially use all the things that we've become very, very good at using in the context of protein folding, in the context of nosy NMR, in the context of all those sorts of things. But we have to be careful because we're measuring a different sort of thing, and learning how to deal with contact probabilities as opposed to distances is an important thing to do. And the last issue is that humans are diploid, and they have repeats. So I've kind of pretended that after we're done sequencing, right, I can read off the blue piece and I read off the red piece, and I know where they came from in the genome. And That is true if I had a kind of haploid genome that had no repeat structure. I would always be able to map things back. But for instance, diploid genome, you have a repeat structure, right? Every chromosome repeats twice. And then you have repeats all over the place. So lots of these reads are not necessarily mapping back unambiguously. And so dealing with the fact that things don't necessarily map back unambiguously is a very tricky thing. There's some very, very extreme cases of this. For instance, the aplasia giant neuron literally contains 200 petabytes of information in a nucleus which is smaller than 1 cubic millimeter. So it's much, much better than an iPod, for instance. But it does this by having 500,000 copies of its genome. So on the one hand, you could do amazing single-cell experiments on such an organism. But on the other hand, you would have to deal with this problem in a very, very serious way. Anyway, so this is what one of these matrices looks like. This one was generated by Suhas Rao, who's an insanely talented student in my lab. He's really just unbelievable. Anyway, so we made this map. Just to give a sense, right, this is, in some sense, a matrix that has of order quadrillions of entries. So even just figuring out how to visualize these sorts of things can be quite tricky. Anyway, you can't really get much of a sense of anything at this level of resolution. I just want to point out that what you're looking at along the diagonal are the individual chromosomes, right? Those are living along the diagonal here. And then all of this stuff that's kind of far off the diagonal, not these diagonal blocks, those are these interchromosomal spaces representing contacts between different chromosomes. Just to zoom in a little bit, so let's look at, for instance, chromosome 8. Here's a map of chromosome 8. And just to give you a sense of what you need to do in interpreting some of these sorts of maps. So suppose I'm interested in locus A. So how often, the first thing to sort of do is think about how often does locus A contact itself. And you can see that it contacts itself quite often. That makes sense. Locus A is very close to itself in the sense that I am very close to myself. So it should be quite intense, and in fact it is. And that's not just true for locus A, but it's true for every locus on this chromosome. In fact, on every chromosome of a successful high-C experiment. So what you see is this very, very intense diagonal. Yes?
So you mentioned the diploid problem.
Mm-hmm.
So what is this chromosome 8 with itself? Or with
yes, this is chromosome 8 with itself.
Or
the
copies, or?
Yeah, so the vast majority of the interactions that you're seeing here are intrachromosomal interactions. There is a small population of interchromosomal chromosome 1 interactions. There's two possibilities in terms of thinking about this. One is it is in principle possible that sister chromatids tend to be spatially very close to one another, closer than the average pair of chromatids. That could actually be a significant confound for high C. Thus far, the microscopy evidence doesn't suggest that. The microscopy evidence suggests that what's actually going on is that sister chromatids are not particularly proximate to one another. Therefore, it's reasonable to believe, based on that, that the likelihood of contacts between two copies of a chromosome, say chromosome 8, is much, much lower than the likelihood of a contact between things that are on the same copy of chromosome 8.
You see that in the haploid like these?
So we have not done haploid experiments yet, but this is something that we would like to do. I'm not sure that we would be able to get at this particular question in a haploid lineage, though. So notice we have lots of these diagonals, lots of these local interactions, which lead to a very intense diagonal on essentially every chromosome in every genome in a successful such experiment. But you have lots of other structure as well, right? You have these polka dots in the upper left. You have this kind of starburst pattern in the lower right. So there seem to be features that are interpretable here. Of course, you see less intensity between, say, locus A and some locus B, still less intensity between locus A and locus C. So there's a lot of heterogeneity and sort of patterning on these maps. So I just want to show you results at 3 different size scales inside the nucleus. We're going to start with things that everybody knows about, essentially the scale of the entire nucleus as a whole, and talk about chromosome territories. And then we're going to zoom in by essentially a factor of 10, think about structures that live at the scale of about 10 megabases instead of chromosome territories, which are each chromosome is about 100 megabases. And then finally, we'll zoom into the megabase scale and talk about potential structures that live at the scale of about 1 million base pairs. So again, zooming in by a factor of 10. So let's think about what we would expect this to be like, right? How should chromosomes organize themselves? Well, if I were to take some noodles and put them in a pot, all right, let's say I dye one of the noodles green. I put that in a pot. And then I set up some sort of time-lapse photography over to the side. And I let the noodles kind of do their thing as I'm boiling them. What I would expect is to see sort of a little bit of green all over the pot, right? That's what I would see if I saw the time-lapse photography, because this thing is oscillating all over the place, and it creates this greenish hue. What I wouldn't expect to see is that the green chromosome kind of formed a small little blob somewhere in the middle of the pot, right? That it was kind of antisocial in this respect, and kind of secluded itself in a small corner. That would be physically bizarre. In fact, though, and there's a picture of an idea about how the genome folds up at the scale of the whole nucleus. See on this cover of Genetics by Hartle and Jones. This is precisely what you see, right? Is that the individual noodles are not completely interlinked and totally intermixed, but in fact they're actually spatially segregated. And that's quite interesting. So here, each of the chromosomes is in different colors. So that's quite interesting. This has been known for some time. That's an interesting mystery, actually, as to how this managed to persist in the way that it does in the cell. It's not quite obvious that such a thing should. So we thought we ought to be able to see this sort of thing in the Hi-C data. And in fact, if you plot contact probability, the likelihood that two loci are in physical proximity as a function of one-dimensional distance, what you see is that the contact probability decays very, very rapidly and eventually hits a plateau. But the interesting thing about this plateau is that the plateau is several-fold higher than the likelihood that two interchromosomal loci are interacting. So what that tells you is that even if two things are 200 megabases apart in 1D, they are more likely to be in immediate physical proximity than things that are in different chromosomes. So we interpret that to imply, to be consistent with the existence of chromosome territories. Just to give you a sense of what this looks like under the microscope, here are the two chromosome territories of chromosome 7 from a paper of Zink and others. Also, it's known that there's actually some organization to the chromosome territories, in particular that small and gene-rich chromosomes tend to cluster at the center of the nucleus. And that's actually what you see by Hi-C as well. If you look at the number of contacts between different pairs of chromosomes, what you see is that the small and gene-rich chromosomes tend to exhibit more interchromosomal contacts than you would otherwise expect. There's an exception, which is chromosome 18, right? Chromosome 18 doesn't seem to talk to a lot of the other small gene-rich chromosomes. Why is that? Well, it's actually interesting. Chromosome 18 is kind of a well-known exception. It is a small chromosome, but unlike the other small chromosomes, which are all gene-rich, chromosome 18 is a gene desert. And so it's known that it actually doesn't tend to cluster at the center of the nucleus quite as well as the others do. And we see that by Hi-C. This is just an image of what these things look like in the nucleus from a paper by Haberman and others in 2002. In fact, folks have actually made efforts to reconstruct three-dimensionally the relative positioning of the chromosome territories from this type of Hi-C data. So that's an interesting sort of thing that you can do and use it to predict the results of fish with some success. So now I'd like to move in to zoom in by another factor of 10 and point out some structures that you see at the scale of the internal structure of individual chromosomes. So the standard way of looking at this type of ligation data going back many years is that let's suppose I've got two loci that I'm interested in, these two yellow guys, if they interact, I should see lots of ligation junctions. But And how does that play out? So you take your observed matrix, you come up with some expected matrix, and then you say, okay, I'm seeing more, I'm seeing less than I expect. So one of the things that we had in analyzing this data was that if you look at nearby loci, these two yellow guys, they shouldn't just form lots of ligation junctions with each other. Something else that you could look at is that they should share lots of neighbors, in the same sense that I tend to know more people in Cambridge, Mass, than, for instance, the folks in Cambridge, UK do. Folks in Cambridge, UK tend to know more people in Cambridge, UK than people in Cambridge, Mass do. Right? So if you are spatially proximate, you tend to share a lot of friends. So what we ought to be able to see is kind of clubs, right? This sort of spatial segregation within the nucleus by looking for shared interaction patterns. Now, how does that play out in practice? So I start with my raw counts. I divide by my expected matrix, and I produce an observed over expected. Then what I look at is I've got, let's suppose I'm interested in locus 1 and how often it interacts with locus 2. So I can look at how similar are their interaction patterns. In fact, I hope that folks can see. Can you raise your hands if you could just see that locus 1 and locus 2 have very similar interaction patterns? Perfect. So anyone who wasn't raising their hand should just identify someone who was raising their hand, and hopefully that person will testify to the accuracy of my statement at the end of the talk. This is a very, very strong correlation, right? Wherever this is overrepresented, the other one's overrepresented, and vice versa. It's a strong correlation. We'll mark it with a bright red dot. Let's compare that instead to locus 1 versus locus 3. Well, that's interesting, right? Wherever locus 1 is enriched, locus 3 is depleted, and vice versa. So they have exact opposite patterns. There's a strong anticorrelation. We can mark that with a bright blue dot. And it turns out, right, this is very slow, so let's just do this everywhere. This form of correlation analysis really brings out a great deal of structure in the data. It's very, very powerful. In fact, you don't even get quite as good a sense of the power of this technique from this example because there was so much data in the original matrix. Anyway, the positive correlations are shown in red, the negative correlations are shown in blue, and the general absence of correlations is shown in black. Now you can see that the structure that you're seeing in these correlation maps is very similar to structures that you were seeing earlier, but it's much better defined. To the extent that you had data sparsity before, it gets filled in really nicely. And now, actually, most of the pixels are statistically significant. That said, they're not quite independent, so what exactly you do with that is not clear. But you really get much more robust statistical results from this kind of approach. So the most striking thing about high-C maps, especially when you do this, is that there's obviously two opposing patterns, right? Here we've got two 10 megabase long runs, and they look very, very similar in terms of the interaction pattern. Here's two more runs, 10 and say 5 megabases. They've got the exact opposite pattern, and that's pretty much all you see. It's all you see on chromosome 14, and it's all you see on any other chromosome. So they make these just very, very beautiful kind of psychedelic patterns, which I've always wanted to make a rug or something like that looks like this. So anyway, so they make these just very, very beautiful patterns because it's just these two opposite patterns. So what's going on? What's an interpretation of that? And our interpretation of this is that what you're seeing is a segregation of the genome into two compartments, what we'll call compartment A and compartment B. Right? So there's lots of contacts within compartment A, lots of contacts within compartment B. There's not a lot of crosstalk between compartment A and compartment B. So, right, this is kind of a claim, and so it seemed reasonable to try to verify that claim. And I don't want to get into the details of this, but it turns out that actually when two things are in the same compartment, even if they're very, very far apart, on the genome, they do seem to show this statistical tendency to be closer together. They're not right on top of each other, but they do show a pretty pronounced tendency to be closer together. In 3D, if they're in the same compartment, even if they're much, much further apart in 1D. So the question is, what do these compartments correspond to? Kind of a simple linear algebra trick. You can do this at higher resolutions. This is a 100 kilobase map from the same data set. So by a simple linear algebra trick, if you compute the principal eigenvector of this, it points up whenever you're in in one of the compartments and down whenever you're in the other. So now you have this one-dimensional thing. And the convenient thing about having this one-dimensional thing is that now you can compare it to all kinds of other one-dimensional things. So for instance, where are the genes in the genome? Where's like K36, an active histone marker? Where's DNA's hypersensitivity, mark of open chromatin? It's actually very, very clear that whenever this eigenvector is kind of pointing up, whenever you're seeing one pattern in the high C matrix, it's living on top of the places where the genes tend to be, the parts of the genome that are actively transcribed and show K36 trimethylation, and the parts of the genome which are accessible to DNA. It's the open chromatin. So what you're seeing is really segregation, not just between compartment A and compartment B, but you're really seeing segregation between open and closed chromatin, genome-wide. And this is actually a dynamic thing. So this locus, actually, I used to like to call the Norwegian locus because I don't really know geography, and I very erroneously believed that it looked like the flag of Norway. That's comically wrong. Anyway, I was very struck when we redid this experiment in a different cell line, K562, that all of a sudden this Norwegian locus, which was like very beloved, had disappeared. Right? It went from kind of looking like this to looking like that. It looked like an ordinary checkerboard locus. So what's going on there? Well, you can see that originally, this kind of locus, which is about a region of about 5 megabits, is kind of oscillating back and forth between the two compartments. And then all of a sudden, in K562, it's kind of decided to commit. It's committed to the open compartment. And what you're seeing before is the little bits that were in the open compartment did correspond to little peaks of DNase accessibility. Now you've got this broad piece of open chromatin, and you see that in the orthogonal DNase accessibility, right, that this is in fact open chromatin. So what you're seeing is actually that gene activation is accompanied by spatial migration from one of these compartments to the other. So the last thing that I wanted, and in fact this turns out to actually be pretty ubiquitous, so now there's more actually work out of the Decker group showing that you can see this in mouse, this sort of compartmentalization. You can also see it, some work out of the Tanai group and others showing that you can see this in Drosophila. So this is actually seems like a motif that you see throughout a wide range of higher organisms. And it's an interesting question as to when this sort of structure started to exist. And if you think about it, it's kind of like a new type of mark, right? Most of the existing marks, right, were like, if I want to emphasize text, what I do is I maybe put a box around it, or I make it bigger, I make local changes, I make biochemical underlines, biochemical boxes, etc. Of course, the most influential thing that you can do when trying to emphasize a piece of information, say in the newspaper, is not to put it in a box, but to put it in a particular configuration with respect to the fold, right? So here, we've put something on the front page, right? And that's going to make a bigger difference in terms of people seeing it than any box that we might put around it. So in some sense, what we're seeing is a spatial epigenetic mark, right? The genome appears to fold into some way that makes the parts of the genome that are of interest to a particular cell type accessible. So lastly, in the last couple of minutes, I just want to talk about some of the models that we and others have been working on about structure at the megabase scale. So we're zooming in again, and now I'm going to consider the problem of what do 1 million letters of the human genome look like? So there's two different ways of thinking about this problem, right? There are two kind of orthogonal different ways of thinking about this problem. One is, well, the genome is 1 million genetic letters, and really as a physicist, there's no moral difference between any of these genetic letters, so you can just think of it as a polymer. And of course, there's a whole branch of physics dedicated to what polymers do, and so you should think of, so we should be on the lookout for interesting polymer physics. An orthogonal way of putting it is actually well phrased by Leonid, who's sitting right here, is that a genome is like a library for a cell. So it should have the properties of a good library. It should be compact. All the books should be in one place. It should be organized. You should be able to find the book that you're interested in finding. And it should be accessible when you find the book. It should not be behind glass. You should be able to pull it off the shelf, leaf through it, and put it back. So this is, in some sense, the way that one might think of this if you were an information scientist or a librarian, right? The critical thing is the differences between the bases, the information that they contain, and organizing that information. So I would like to try to do is think about how we can reconcile these two very disparate ideas in a model. It's not meant to be a final solution or any sort of thing, but just a model that is more refined perhaps than the types of models that have been thought of before. So one last methodological preliminary. So every curve, when you have a curve, it has a contact probability function. What do I mean by that? Well, every once in a while you fold up, curve is basically a one-dimensional thing, fold it up into higher dimensions, and every once in a while these things come into contact. And those contacts are associated with distances. This is a 1, 2, 3 distance contact. This is a distance 13 contact. This is a distance 213 contact. You can see the coloring used here. It ranges from blue to red. And so you can always get a sense of how long distance the contact is by how different the colors are. Anyway, you can make a table of how contact probability scales with one-dimensional distance. You can make a plot of this, and you can discover that this plot has a particular shape. Now, for the human genome, it turns out that you can do this. And the human genome, in fact, is actually the only polymer for which you can do this, which is an interesting sort of thing, because the human genome is essentially a colorful polymer, because all the different pieces of the human genome look slightly different because they have slightly different bases. Anyway, so you can plot contact probability as a function of distance. And what you notice, which is quite striking, is that between about half a megabase and around 7 megabases, you see a pretty pronounced scaling, which is pretty close to -1. Now, that's kind of, that's interesting, right? Why are we seeing that? It's a power law. In general, those can be very, very hard to explain, but in polymer physics specifically, power laws actually have a very robust tradition. So it was reasonable, when I originally observed the scaling, to just go back to my old polymer physics textbooks and try to figure out what was going on. Anyway, possible folds. Well, the classic way that a polymer condenses, all the textbooks tell you about, is, it folds up into a structure called the equilibrium globule. And this is very, very dense. You can see it's compact, but it's very badly organized. It's a big mess. And it's accessible. No, it's got lots and lots of knots. And the slope, actually, that you would get for this, initially I was like, oh, this is very, very cool. But actually, I pointed this out to a number of different people, including Leonid and his graduate student Maxim. Leonid is right here. And they pointed out that actually an equilibrium globule would not give a scaling of -3/2, which I found quite depressing. So what I did in response to being very depressed about that is that probably what many of you might have done, which is that I read many, many papers on polymer physics, and that turned out to actually be an excellent cure for acute depression. In particular, this one paper by Shura Grossberg and others, Sergei Nechayev and Shura Grossberg, Sergei Nechayev, and Eugene Shakovich, in 1988, where they suggest that a very, very long polymer, when it collapses, could get arrested into this state called a fractal globule, and might stick around in this state for some time. Now, the fractal globule is interesting. It's highly dense, but it's also extremely organized. Right? You can see these big monochromatic blocks, and it's accessible. It ought to have no knots, according to theory. And the simple back-of-the-envelope calculation that I did suggests that it ought to give a slope of -1. And in fact, that has been borne out by both subsequent theory and some simulations. So initial work was done in collaboration with Leonid Murnin. I'm going to skip this. And now we've just been doing a whole bunch of other stuff in this space. And I just wanted to show you a couple of the types of simulations that we've been doing. So, of course, the fractal globule looks kind of similar to this chromosome territory structure. Right? It's kind of appropriate, right? You've got this modular design. It kind of looks like it ought to be the same as this thing, but it's actually representing something quite different. Internally, you have these compact subglobules as opposed to these diffuse knots that you would get with, say, an equilibrium globule. Of course, it's very hard to draw. I've been showing you beautiful pictures like this. This was actually created by Miriam Huntley and my group earlier this year. In fact, we didn't have pictures of this type. At the time, this was kind of the state of the art in drawing fractal globules. And so initially, in collaboration with Leonid and Maxim, and now independently, my group has been doing a lot of folding of polymers to see what it produces. And it's actually very easy to get interesting results out of this. So for instance, here's a random configuration of a polymer. This was work done by Ashok Kutkowski in my group, together with and he worked actually with Najib Tarazi building the visualization pipeline that we use. So this is a collapsing polymer. You can see it forms these little beads. These little beads kind of collapse into larger beads. So you've kind of got beads of beads. And these beads of beads kind of collapse into still larger beads. So you've now got beads of beads of beads. And these things kind of continue to collapse until you've eventually got this sort of bead of beads, which is a fractal globule, kind of a globule of globule of globules. It's a very robust result. You can get these in many different ways. So anyway, that's good. Turns out these things give contact probability scalings that are pretty close to -1. One can also simulate equilibrium globules. So simulation is just being lots of stuff being packed into a very, very small space, and then gradually reinflated. And this particular thing is actually following the original work done together with Maxim and Leonid, and now re-implemented a bit faster. So anyway, we can do all kinds of these simulations. You can see the sort of theoretically expected behavior, more or less. Fractal globules are unknotted. So for instance, if I treat this as a big spring and allow the fractal globule to collapse, although it looks very complex, it actually collapses right down to an unknot, which is really convenient. And equilibrium globules do not do this. So let's allow that to collapse. It's doing pretty well so far, but then it gets stuck. It's this horrible knot. It just can't get it past this point. So yeah, so I'm sorry. So let's actually, let me show you the initial configuration, because that's a very good point. So one issue, which I should just, point out to just get us on the same page, and I apologize, let me go back. There we go. So this thing is actually an open chain, so we add an ear to it to make it a closed chain. So now we can actually talk about this from the not theoretic standpoint and in a meaningful way. And it turns out then we can calculate all kinds of knot invariants of these sorts of things and talk about them in a slightly more rigorous way. Now, the lack of knotting actually has an important functional consequence, which is illustrated as follows, right? You can pull on a particular locus of this, right? So here we are pulling on a particular locus, and because it's unknotted, it just comes apart really nicely. And in fact, I can push stuff back in, right? You can do this at multiple sites at once, et cetera. It's actually a modular kind of information design. So it's amazing to think, actually, that the aplasia giant neuron packs 200 petabytes of information, can pack, in principle, 200 petabytes of information into a, less than a cubic millimeter, and yet keep all of it accessible using this sort of structure. So I just wanted to wrap up. We have some ideas about what's going on at these intermediate scales. I wanted to thank lots of people who are involved in this. We're not the A team or the B team, but I at least like to think of us as the C team. In particular, deeply indebted to Louise Williams, especially Nika Van Berkom, for experimental collaboration. Nika was really my main collaborator in all this work. Job Decker for his support, Eric Lander, my advisor, for his support, and for encouraging me to do this as opposed to all the other bad ideas I had. Andy Gnerke for providing a lot of experimental guidance. I mentioned Maxim and Leonid did the very first versions of the kinds of simulations that I'm showing you and that we've been doing in the lab. Some of the simulations that I showed you were done by Ashok Kutkowski, Miriam Huntley, Leon Furtgott, and Najib Tarazi. So what's next? This is just going to be a bit of shameless advertising, but we've had a good run. I've had a good run. I got this paper, which was on the cover, and this paper is on the cover. And this is actually the first paper out of my group, which was also on the cover and led to this thing that now millions of people use called the Ngram viewer. So what should we do next? Well, I'd really like to get people. So I'm looking for postdocs. If you're here and you're the kind of person who, given something that you get sufficiently enthusiastic about that is scientific, are willing to work, up till between 2 to 3 a.m. Past that is more hardcore maybe than I would expect. But then please let me know if you're interested. We're doing a lot of stuff in the space of genome organization. If you're an experimentalist, do molecular biology or 3D fish or things like that, certainly would love to talk with you, or if you're doing computational or theoretical work. Anyway, thank you guys very, very much.
Are there further questions for Erez?
You simulate those, the aggregation and unfolding. What interactions do you actually put in? What is the model?
So it varies. I mean, this is basically.
So I'm sorry, could you repeat the question again, please? We can't hear the questions in the back.
Oh, sure. The question was, what are, essentially just, I kind of just showed this picture and didn't tell you anything about it. What's driving the dynamics? The thing that we most rely on is just Leonard Jones potentials and molecular dynamics. That's kind of the mainstay for a variety of reasons. That turns out to just be the best way to do a lot of things.
Excluded volume
Yeah.
fraction.
Yeah, exactly. So you have, long-range intermonomeric attractions, and then short-range excluded volume. In fact, a student of mine, Asha Kwiatkowski, is here, and we'll be giving a poster on this sort of thing. So you can ask him questions, and he will give you answers. So, and has.
Do the results depend on the parameters?
To some extent, some of the results absolutely do. They also depend very, very, very critically on the size of the polymer that you're simulating. So below particular threshold sizes, some of these types of scalings are actually not meaningful. They depend so crucially on the parameter choice that there's essentially no result there at all. But past certain thresholds, once you get sufficiently large polymers, you can start to make pretty robust statements about their behavior.
In both the fractal globule and the equilibrium globule, are strands allowed to pass through each other?
So, right, physically speaking, right, strands, other than if you have topoisomerase or you're modeling some other stuff, right, they can't. In the simulations that we use to generate fractal globules, that never happens. Simulations to generate equilibrium globules, that turns out to be a very convenient shortcut for generating equilibrium globules. That was actually originally work in Leiden's group that just kind of observed that you could
The
generate.
phantom in the equilibrium, but they're not phantom in the fractal?
There is a phantom phase in generating equilibrium globules. There's no phantomness in generating fractal globules. It would not be helpful, and it would be counterproductive in many, many ways. There's the formation of these kinds of fractal globule-like structures is incredibly dependent on the fact that the strands cannot pass through each other. It's quite fragile. So if you start to allow those strands to pass through each other, you can, make a mess of that quite rapidly.
Your fractal globule has got lots of nice theoretical properties, but as I understand it, there's very little confirmation of these predictions. You've got 3D fish. In your 2009 paper, you have only a handful of those types.
Yeah, so I'll show you quickly if I can. So there's a little bit, or actually the most interesting 3D fish data is this particular scale. This was reported actually long before. So I should say first, I agree. I mean, this is a model. I think, it's a step in right. I like to think that it, has the potential to be a step in the right direction. It's clearly not the ultimate step. And I agree. I mean, right now we're in a state of having a little bit of experimental guidance, but not nearly what we would want. And so, this is, it's generated, I think, some enthusiasm, but it's a little step. And there's this is certainly going to be revised as certain future experimental things happen. Right, it's just clear that this is going to be revised. But I still think it's good in that it helps create a conversation about certain topological aspects, larger scale structure of DNA. I just wanted to make this point about this fish data. This is some of the data that actually I found very, very compelling and referred to in the paper. So it's a plot of genomic distance versus mean interphase distance in generated using several 3D fish data sets, and you see that right around this, kind of megabase threshold. Also, you kind of argue, it's between orders of hundreds of kilobases megabases, it seems to shift to a like 1/3 slope. It was not, and this is actually what you would expect for a fractal globule. But on the other hand, I mean, this is like a scatter plot of a bunch of points on a log-log plot, and they're all kind of old, and there's not enough of them. So being able to do this sort of experiment way, way, way better is crucial. I mean, I think that there's an extent to which, we've done, made some progress like on the high C front. We have to turn back to, and folks are obviously doing this, but the ability to do this experiment 100 times better, right? And in this experiment, you can't really do 100 times better today. So I think it's important to make that effort.
Erez, so the data that you showed at the very beginning, so is this a different cell type that you analyzed? Is it the same data set? So it wasn't clear.
Which the
showed
original genome-wide map?
From your student, sure.
Yes.
Wow. So is it a different cell type, or is it, so what's?
Yes, it's a different cell type. It's a new data set.
Which cell type, and how different is it from the Baker data that you showed me?
So it's a closely related cell type to the work that we did in the original paper, but it's unpublished work. So
maybe we should, in the interest of time, save further questions for lunch and just have a minute or two break. But in the meantime, for those standing in the back, I can count 9 chairs if you're daring enough to sit in the front row, if you'd like to move up during the break. Thanks, Erez.
no problem.